Hi everyone, welcome to the Lymphatic and Immune System series of lectures. In this part we're going to introduce some organs and structures and features of the lymphatic system. Now you might be wondering, what is the difference between immune system and lymphatic system? The quick answer is that the lymphatic system is more of an organ system in that it has specific organs that are part of it, specific tissues that are part of it, and specific kinds of cells. The immune system is a little bit different because even though it's important and it has many roles in our body, there aren't like immune organs specifically or organs specific to this system. Rather, the immune system is the way that our bodies protect themselves given the different interactions between lots of structures and cells of a variety of organ systems. So that's a little bit of the difference between them. So we'll present here the lymphatic system, kind of an overview. Okay, to start, we'll begin with functions of the lymphatic system. It is a place where fluid is recovered. Recall earlier in the vessels and circulation series of lectures that we had the capillary beds, right? You had this place where there's capillaries, and then fluid comes out through the arteriole, it passes through the capillaries, and now it's collected through the venule. Well, at the proximal end, hydrostatic pressure, it favors the efflux of fluid, or the fluid to leave the capillary. And at the distal end, it favors the influx of interstitial fluid. And for reasons that I won't rehash here, but again, you should kind of know why that is the case. All right, about 85% of the fluid is reclaimed at the distal end, which means that there's still 15% of the fluid kind of remaining. Well, that gets dished into the lymphatic system. And it works out really well this way because the lymphatic system is a place where it has a lot of structures geared towards screening for pathogens. And so any pathogen, whether it be a bacterium or a virus or a parasite or a toxin or what have you, if it gets swept through that, the fluid that's continually passing through all of the cells, it will be brought into the lymphatic system where it can be dealt with. Okay, so that's a good kind of design there. And that leads to my next point, that is heavily involved in immunity, protecting your body from invading pathogens. We'll talk more about immunity. There's the humoral immunity and the adaptive immunity, as well as the there's the innate immunity and the adaptive immunity. We'll talk about those later. All right, an interesting one that we generally talk more about when we get to the digestive system chapter, but we should also mention here, is lipid absorption. So it's a little bit curious. If you were to look at a cross-section of something like the small intestine, which is what this is, the small intestine has little circular folds in it, and on those circular folds are these little finger-like projections called villi, which look something like this. So this is one villus. Here you would see the simple columnar epithelial cells, and then this would be the lumen of the digestive tract. Well, generally, the nutrients that we absorb, or the nutrients that we consume, rather, you can put them into different classes, but the carbohydrates generally get broken down through enzymes, and also the proteins get broken down through enzymes as they travel their way through the digestive tract. And then their last kind of thing that they do is they get absorbed by the epithelial cells, and then once they're absorbed by the epithelial cells in the digestive system, the epithelial cells will kind of actually export them into the basal side of the epithelium, where it can then be absorbed by capillaries that are waiting right underneath. So most of the nutrients that you obtain through your food make their way through the epithelium and then into the capillary beds of your digestive organs. Now, what's kind of interesting is that lipids are sometimes subject to a different pathway. They'll come in through the epithelium. They usually can, diffuse in because they're lipids, so they can diffuse through the hydrophobic regions of the plasma membrane, and they will find their way into a lacteal. Lipids end up, merging with some proteins and forming pretty large molecules. molecules so large that they're not able to seep into the small cracks and crevices and holes of the capillary beds in the villi. And so they have to go through a lymphatic vessel, which is called a lacteal. And they'll eventually meet up back in the blood, but not before they've first traversed through some pathways of the lymphatic vessels. So that's kind of an interesting role there, also the lymphatic system. Okay, the components of the lymphatic system, there's the lymph and the lymph vessels, there's the lymphoid cells, the lymphatic tissues, and the lymphatic organs. Kind of a smattering of different types of structures. This picture shows kind of a summary of all of these things, and we'll go through and we'll talk about these in a little bit more detail here in the following slides. So first up is lymph and lymphatic vessels / circulation. This is a pretty great little illustration here in the book showing a capillary bed. So here's an arteriole, here's a venule. There are all the cells right here. Okay, and it shows that fluid leaves out of the proximal end of the capillary bed, as we recall from our vessels and circulation lectures. And what's interesting, and what we haven't talked about yet, is the fact that there are these interesting little capillary roots, almost. They're lymph capillaries, which kind of interweave through the capillary bed. And whereas a blood capillary is kind of a continuous branch from arterial to venous side, the lymph capillaries are kind of like an origin point. They're kind of like a dead end. They don't really link into from one place to the next. They're more of a starting point, if you will. So that's what makes lymph capillaries unique. Now, as soon as we have fluid leaving the capillaries, it's no longer called blood anymore. Now it's called interstitial fluid. The interstitial fluid is the fluid that exists between all of the cells. And then when it gets any fluid that doesn't get reclaimed by the distal end of the cardiovascular capillary bed, that fluid will likely find its way into the lymphatic capillaries. And once the fluid, which was interstitial fluid, is now inside the lymphatic vessels, now we call it lymph. So lymph is very similar to interstitial fluid. It's very similar to blood plasma, the fluid that's inside of blood, with a couple of exceptions. But that is, when you hear the word lymph, that's what it is. It's fluid that's left the cardiovascular system and now has been taken up by the lymphatic system. Here's kind of a picture that really shows the microanatomy of what's going on here. So if you take this one little lymphatic capillary and enlarge it, what's interesting about these lymphatic vessels is that they have endothelial cells that overlap one another and create kind of these little microscopic valves. So when the pressure builds up in between the cells, the interstitial fluid, when the interstitial fluid has a high pressure because it's building up, then it will actually pop these valves, these little cells apart, and then the fluid will kind of rush in, and then it will kind of seal again. So in that sense, it kind of promotes a one-way movement of fluid from the origin, which would be all of these these are called the blind ends, because they just kind of stop. They're like dead ends. So it promotes one-way movement of fluid into the blind ends of the lymph capillaries and then through the lymphatic vessels. Additionally, lymphatic vessels have valves very similar to veins. So kind of a couple different special properties here, the lymphatic vessels. You can see here the fluid that is outside of the capillary is called interstitial fluid, literally meaning fluid in between cells. You can see, collagen as representing the extracellular matrix between these cells. And then once the fluid enters, now we call it lymph. Well, all of the fluid that's been kind of taken up by those lymphatic capillaries gets funneled into increasingly bigger and bigger diameter vessels, and then you eventually get these bigger lymphatic vessels that almost look kind of like the cardiovascular vessels. And then ultimately all of these vessels that have picked up lymph from capillary beds throughout all of the body are going to bring it back to the heart. And there's two main ducts that will carry that fluid, which has been kind of sucked up from the rest of the body back to the heart. There's the right lymphatic duct, and there is the thoracic duct. The right lymphatic duct will collect all of the lymph that has been picked up in this quadrant of the body, and then the thoracic duct will pick up lymph that's been collected in the rest of the body. And there's this actually bigger thoracic duct right here where a lot of it pools into before it then passes in. The point of insertion for these lymphatic vessels is right at kind of the bifurcation of the jugular vein, the internal jugular vein, and also the subclavian vein. So here this would be brachiocephalic vein, and then we have this right here is the subclavian vein, and right past the internal jugular vein is where these ducts enter on both the right and the left side. So then it can rejoin the cardiovascular system, and ultimately no fluid is really on the net lost out of the cardiovascular system, which is good because you don't generally want to lose fluid out of your cardiovascular system. Okay, there are a couple of diseases that can affect the flow of lymph, and we've already talked about this earlier, but just to summarize again, lymphedema is a condition where there is swelling due to a blockage in the lymphatic vessels. Here's a picture of lymphedema. So you can see in this extremity it appears relatively normal. Here this appears more swollen. The reason why it's swollen is because the fluid that has left the capillaries of the cardiovascular system has not been reclaimed, and so it instead pools in the tissues of this lower extremity. Now, if this gets really severe, it can start to cause a thickening and kind of almost like a leathery type nature to the skin, and it can actually get pretty severe, as you can see in this particular image. And when it does get to that severe level and the skin gets rather thick and hard, it's called elephantiasis because the skin becomes like that of an elephant. Anytime I see, if you're looking at pictures of severe lymphedema and it doesn't, pull on your compassion, it's just really, really sad to look at these types of images and think about the suffering that some people are going through with this condition. Lymphedema has a couple different causes. The first are characterized as primary causes, and this is generally genetic type causes. There's a whole bunch of then secondary causes, and secondary causes, some of the more common examples of reasons why this disorder, lymphedema, would occur is one of the main reasons is cancer. So what's good about these little gaps between the endothelial cells of a lymph vessel is that they're big enough to even allow cells through. And that's great because a bacterium or a macrophage could find its way through there and into the lymph and then be on the road to identification and destruction. But kind of a caveat of this opening is that it's also big enough to allow cancer cells to get through and thereby allowing cancer to spread rapidly through regions of the lymphatic vasculature. And so this is a picture showing a kind of a cancerous tumor in a cell that has metastasized or broken off and spread, and it's been able to, they're enlarging this to show you that it's made its way through the two endothelial cells of this lymphatic vessel and then into the lymph. Well, obviously, when that happens, it has to be removed if it can, so either through surgery or through radiation or chemotherapy. It might be the case that certain regions of the lymphatic vessels are destroyed, and then they consequently don't drain that fluid back to the heart, so it can cause lymphedema. Another cause is a little thread-like worm. There's a variety of different worms that could be introduced to the body. This is a picture showing a little thread-like worm inside the lymph. If the blockage of lymph is occurring because of a parasite, such as the one you see here, it's called lymphatic filariasis. Okay, moving on. The cells that can be found in the lymphatic system are the following. Recall, here's our scheme showing the differentiation of a hematopoietic stem cell into all of the different lineages. Remember that you can characterize all of the lineages into either a myeloid stem cell lineage or a lymphoid stem cell lineage. Well, we're going to focus here on the lymphoid stem cell lineage. Specifically these cells. We have the natural killer cells, the NK cells, the T lymphocytes, and the B lymphocytes. We're going to talk at great length about these. Just real quickly though, I'll introduce them because we'll probably talk a little bit more about these in the following slides. Remember that a B cell is a cell that makes antibodies. And when a B cell becomes activated, it can differentiate into a plasma cell. And a plasma cell can really make antibodies. Plasma cells are kind of like the champion antibody making cells. The T cells, there's two different kinds depending on kind of the different molecules on their surface of the cell and the role that molecule plays. Some of these lymphocytes have a molecule on their surface called CD8. Some of these lymphocytes have a molecule on their surface called CD4, and there's probably actually a variety of other types of lymphocytes with different unique CD8 molecules on them as well. CD is kind of a class of molecule that is used to denote kind of an immune special protein of some kind. So there's lots of CD molecules. There's CD40, there's CD57, there's CD8, CD4, lots of different ones. But in this class, we'll just focus on these two because they help distinguish two main classes of lymphocytes. The CD8 cells are sometimes called cytotoxic T cells, and the CD4 cells are sometimes called helper T cells. The cytotoxic T cells, as the name suggests, cyto meaning cell, toxic meaning destroy, they destroy cells, and specifically cells that are abnormal or infected. The helper T cells will either help to turn on a CD8 cell or they'll help to turn on a B cell. So that's kind of what they do. And then the NK cells are usually more known for destroying cancer cells. We'll talk at great length about the B cells and the T cells here in the next lecture, but for now, just kind of keep in mind the two different kinds. Okay, lymphatic organs. We can differentiate lymphatic organs into primary and secondary lymphatic organs. A primary lymphatic organ is one that produces B or T cells. A secondary lymphoid organ is one that doesn't necessarily produce the B or T cells, but it's a place where with, I guess, lots of B or T cells, to put kind of informally. It's also a place where B and T cells could divide, and here's where you have to kind of keep in mind a different kind of a differentiation scheme here. By produce B and T cells here, I mean that it creates new kinds of B and T cells. There are billions and billions of kinds of B and T cells. Each B and T cell is that comes out of the primary organs are unique, almost like a snowflake. And so this is where the unique cells are made, but in the secondary organs, there's still cells made here, but they're usually clones or duplicates of those cells which have been made in the primary organs. So even though there's a lot of cells being produced in the secondary organs, they're all copies or clones of cells that were originally made in the primary organs. So keep that in mind. That will help you distinguish between primary and secondary lymphoid organs. Here's our little scheme of all the different organs on the lymphatic system. The red bone marrow and the thymus are the main primary lymphoid organs. The thymus is this little gland here that sits above the heart, and as the name kind of suggests, T, this is a place where the T cells are made. Now, I guess I have to be a little careful here. The T cells are also made in the bone marrow, same as all of the other white blood cells, but then they'll usually travel up to the thymus where they mature into a T cell. So that's the thymus, and then the red bone marrow we've already talked about. That's the place where hematopoiesis is occurring. So all the blood cells are made here, including the B cells. Now, you might be tempted to think that B cell stands for bone marrow. You know, the B in B cell stands for bone marrow, and the T in T cell stands for thymus. The B actually stands for bursa, which is an organ in a bird in which they were first identified, but it kind of helps as well because we can think of it as standing for bone marrow. And then T does stand for thymus. What's interesting about the thymus is that it gets kind of bigger and bigger and bigger as we grow, and then after puberty it starts to atrophy or get smaller. And it kind of gets smaller and smaller and smaller later on in life. And some think that that's partially the reason why you're a little bit more susceptible to illness in old age, along with, you know, a whole bunch of other reasons. But at any rate, kind of an interesting aspect of the thymus there. Now, while the thymus is more well known for being a place that T cells go through their training, one might also find B cells in that organ as well. So just to clarify. Okay, the secondary cells organs then are the following. There are the lymph nodes. There is the spleen. There is this stuff called mucosa-associated lymphoid tissues, and of which here are some examples. And we're actually going to go through and talk about each of these things in a little bit more detail, but just briefly here, the lymph nodes. These are these little kind of bumps that you see enlarged on the lymphatic vessels. The spleen is this organ right over here. And then the mucosa-associated lymphoid tissues, those are found in a lot of places. The tonsils are up kind of by the throat. The Peyer's patches are going to be found kind of in the small intestine. And then the appendix is this little thing that kind of dangles off right here, right next to where the large intestine is. It's also going to be a place that has these things, which we call malt. We'll talk more about those in a second. Okay, the lymph nodes. Here's a picture of a lymph node. There's about 500 to 600 of these in the average adult. Some sources will say a little less than that, like more like 450, but still a lot, right? And their main job is to filter lymph. So about 99% of the lymph that is collected will pass through lymph nodes. So anything that is trapped in the fluid surrounding cells and makes its way into the lymphatic vessels will pass through likely a lymph node. Inside that lymph node, there are a lot of cells which can activate an immune response. So you can see this diagram is trying to show you a bunch of cells in here. These cells are like really big, drawn to scale. They'd actually be like microscopic here if this was drawn to scale, but in essence they're just trying to illustrate how many cells end up finding their way into this lymph node. And this is a place where B and T cells and macrophages can all come together, and those are all important things that need to happen in order to generate an immune response. And we'll talk all about that in the next couple of lectures. I think the next lecture. Okay, the anatomy. They have afferent and efferent vessels. So afferent vessels are going to be ones that carry lymph into the lymph node, like this, and there can be many afferent vessels. And then there's usually only one efferent vessel, sometimes two, but oftentimes one. And the main idea is that there are more afferent vessels than efferent vessels, so it creates kind of a bottleneck, which is going to force fluid to slowly pass through here and give that fluid plenty of time to cycle through a whole parade of B and T cells. Surrounding this lymph node is a fibrous capsule made out of fibrous regular connective tissue. Then there are these little projections that create little partitions between these little segments, which are called the nodules. The cortex is going to be a place on the outer side, the outer portion of the lymph node, and that's going to be a place where there are these little collections of cells called follicles. So each one of these things is a follicle. Now a follicle is going to be a place where there are many, many lymphocytes. Generally how it works is there's a thing called a germinal center. I don't have that written down. In a germinal center is a place where there's a B cell that is cloning itself, preparing for war against a pathogen. We said that there are billions and billions of different kinds of B cells out in the body. Each one of those B cells can detect and bind a specific pathogen. And if some virus enters my body and there's the specific B cell that matches it, it will be selected to divide. And a great place where it can divide and start creating its army is in the lymph nodes. So when you see a germinal center where there's tons of cells dividing, that is a B cell which was made originally in the bone marrow, but it's now cloning itself in what's called the germinal center. So usually this is B cells cloning themselves in the germinal center, and then usually surrounding that germinal center are the T cells on the surface. And why it arranges in this way, I don't exactly know. I don't know why the B cells are usually in the germinal center and the T cells are surround it and not vice versa. If you ever find out the answer, send me an email, let me know. Okay, so that's B cells and the T cells in the follicles in the cortex. And then in the medulla, this is kind of this inner more region here, is where there's a collection of all sorts of cells, but in addition to the B and T cells, a lot of the macrophages are often found here. Macrophages usually don't hide out with the B and T cells in the follicular center, so they'll kind of pass through and collect right in this region, and then they'll continue on. Again, as you can see from it, it's a great place for cell mingling, and that's going to be very important to activate an immune response, and you'll understand why in the next lecture. Here's a slide showing a lymph node, and you can see here's like the different nodules, the different regions in the cortex, and these little lighter areas would be the germinal center. They're a little bit lighter in color because they're fresh cells which have divided, so there's a little bit more space between them, so it looks a little bit lighter in color. And then you can see all of the kind of the T cells that are surrounding them in these big follicles. And then in the middle, this is where fluid kind of collects before it goes outward. You can't really see the afferent and efferent vessels, and I wonder if that's because they're actually kind of coming in like this, kind of from the bird's eye view, and then collecting here and then leaving out. That could be why. Okay, in addition, there's a bunch of little fibers that are going to be sprawling throughout a lymph node, and this whole mesh of fibers is called the reticulum, and it's actually a place that allows, it's a network that allows cells to anchor themselves into the lymph node so that they're not swept through with all of the lymph that's flowing past. Okay, on to talk about the mucosal associated lymphoid tissues. These are just first we should talk about what a mucosa is. A mucosa, if you don't recall what this is, it's pretty simple. It's just an epithelium, so any epithelium in the body that has mucus on it. So if a mucus is on an epithelium, we call it a mucosa. And remember that an epithelium is usually a place where there's boundaries in the body, places where the body could be exposed to foreign things. So oftentimes any pathogen or bacterium that has to get in the body has to first travel through some sort of epithelium. And so in places that are more subject to pathogens infiltrating the body, such as the throat or the nasal cavity, there are these little bundles of cells, B and T cells, inside lymphoid tissue just underneath the surface of the epithelium. So this would be an example of a mucosal associated lymphoid tissue. The big examples here are the tonsils. There are three types of tonsils. There is the palatine tonsils, which exist right underneath kind of the palate. There's the lingual tonsils on the back of the tongue. And then there's also the pharyngeal tonsils. And I won't go into a whole bunch of detail here, but just generally knowing there's three types of tonsils, You can see these are kind of inflamed palatine tonsils on either side of the uvula here at the back of our palate, soft palate. And it's just a great place to have extra B and T cells waiting, because, you know, if you breathe in a virus and it's kind of passing through and it brushes up against this mucosa, the mucosa can, you know, the B cells and T cells right underneath the mucosa can destroy those pathogens. And then similarly, anything we eat is going to pass through not only these, but also there's a little place where food and things can settle into this little crease, and that would be a place where there's B and T cells there to activate immune response towards any of those pathogens. So a little extra protection here for these openings, these very significant openings to our body found up in the nose and mouth. Okay, the Peyer's patches are the same deal, but not in the pharynx. They're down in the small intestine, specifically the more abundantly in the ileum of the small intestine. The ileum is the last branch of the small intestine before the colon. There's tons of bacteria in the colon, and so it's kind of a place that protects bacteria from getting out of the colon and proliferating too abundantly in the small intestine. You maybe have heard of the condition known as SIBO, which stands for small intestine bacterial overgrowth. It's a condition where there's too much proliferation of bacteria in the small intestine, and that would happen more frequently if the Peyer's patches were not there. The colon is a place where there's tons of bacteria. There should not be a whole lot of bacteria in the small intestine. There will be some, but not nearly as much as in the colon. And the Peyer's patches help kind of protect the small intestine from the attacks of, you know, potential attack of bacteria. Here you can see the villi, or these are the circular folds in the villi on them. And so this is kind of the surface of the wall of the small intestine, and then just underneath that there are these little garrisons of B and T cells, and these are all called mucosa-associated lymphoid tissues, little lymphoid tissues full of lymphocytes just underneath the surface of the epithelium. All right, the spleen is another lymphatic organ, and one of the things that's confusing here is that whereas the lymph nodes will filter, you know, 99% of the lymph, the spleen, while also a lymphatic organ, does not filter lymph. It filters blood. So keep that in mind. This is for blood, not lymph. And blood also has the tendency for pathogens and things to get in it. And so it also needs a special organ to help protect it and keep the blood clean, quote unquote. So in this organ, blood will come into the spleen. The spleen's pretty spacious. It's got the reticular connective tissue, which is a little bit more spacious of a tissue, allowing lots of fluid to pass through. So there's lots of room for cells to migrate through. And all the spleen is divided into regions which are called white pulp and regions which are called red pulp. Red pulp is a part of the spleen that has a lot of red blood cells in it. White pulp is a part of the spleen that has a lot of white blood cells in it. And so you can see that there's going to be a mingling with white blood cells and red blood cells. In a slide here, you can see these little darker purple staining regions. This would be the white pulp, because you're seeing, you know, some more of the nuclei of the white blood cells. And then the red pulp would be all of kind of the regions of the spleen that are surrounding the white pulp. All right, that's going to wrap it up for just a quick presentation of some of the structures that one might find in the lymphatic system.